Hello, this is Taras Viktorovich Pluskin from the University of Florida. This is a brief summarization talk about my research for my graduate thesis here, using microalgae as a nanotechnology and as a nanotechnology to specifically produce polyunsaturated fatty acids, which are essential for all forms of life on this planet. Why have I chosen this as my thesis project? Well, the way I see it, every young person on this planet has a vested responsibility in preserving the three base commodities which sustain all of humanity and the greater ecosystem around us. So fresh water, arable land, and then the production of precious compounds from inorganic materials through photosynthesis. So by preserving all three of these and finding new technologies and new avenues to preserve them, and in some cases, as in the third one, to be able to produce them from new novel sources will be paramount in order to preserving humanity's ability to keep growing and to have us be able to at least keep a stable world for our children, if not a better one. Algae is a nanotechnology in both the classical and modern sense. Since antiquity, algae has been used as a living organism, doing its natural thing in the soil for agriculture and in the water for aquaculture, doing primary productivity. It's been known since antiquity that certain algae exposed to certain conditions will produce pigments or poisons, and these things play an increased role in the world today with mass harmful algal blooms and the exploration of new products from algae as we have new technology and can realize what exactly they were up to this whole time. So algae has been utilized throughout history. In the most primitive sense, you can see apes in the bottom left photo, which are using sticks to harvest nutritious algae, filamentous algae, from small little vernal pools. It's been postulated that these apes are even fertilizing and seeding new pools with this algae. In the right, you'll see the Hercules Powder Company stamp. This is because in the First World War, potash was required to make gunpowder, and the Germans had all the potash mines. So kelp was industrially harvested for potash and acetone production. Kelp is actually a 17th century Scottish word meaning gunpowder. So that's a more primitive use of algae again. Even as early back as the 18th century, agar was realized for its ability to be a wonderful food additive. And then later on in the 20th century, in the 19th century, was used to revolutionize medicine by its use as a novel bacterial plate. There at the bottom, you'll see one that I've constructed and seeded. And so basically, these are countless examples that algae has been used as both a brute nanotechnology, basically being alive and doing its thing naturally without any knowledge. And then as we have new technology, we realize the different products that algae makes, and we've extracted, purified these products, and then used them in ways that the living organism couldn't. And this has created waves of agricultural, nutraceutical, medical, biotechnological, material, all kinds of applications are now being used from algae metabolites. So now that there is this huge swath. So we'll just be focusing on polyunsaturated fatty acids. But there's this huge gosh darn thing of all these applications algae can and will be a part of, and will continue and be a part of new things just like nanotechnology as a whole. So I always refer to these golden fats. What exactly are they? Mostly we're talking about the omega-3 polyunsaturated fatty acids. So fatty acids are fats. You can think of saturated fat, kind of like butter. It's concealed and solid at room temperature. It's very thick. That's a saturated fat, relatively inefficient, causes a lot of inflammation when it's burned for energy, associated with a slew of health maladies, and quite frankly, not something that should be part of the everyday diet in mass amounts. What should be part of the everyday diet is these golden fats, these unsaturated fatty acids. And because of their chemical nature and structure, they have a variety of properties that make them gold and high value metabolic currency in a variety of organisms, pretty much universally beneficial. And that's why we'll see a lot of ecosystems associated and structured around primary producers that metabolize and produce large amounts of these PUFAs, as we're going to be referring to them. So essentially, we have high energy content. When they're burned, they don't produce a lot of inflammation. In fact, they can cancel out inflammation in some organisms. There's all kinds of other, they were first recognized for their nutritional value by a man named Dieberg, who wanted to know how native Alaskans could consume so much whale blubber and yet not suffer from cardiac maladies that hurt and were killing people on the mainland. And what they were finding was that when contextualized with high levels of DHA and EPA, that solid fat was more or less becoming liquefied. So there's infinite positive applications of these polyunsaturated fatty acids. So in marine organisms, they cannot be synthesized de novo in many organisms. So when you're rearing marine larvae, many times you must rear them with live feeds that have been enriched with these polyunsaturated fatty acids. Many times that's done by feeding them microalgae directly. Because if they don't get those polyunsaturated fatty acids, they will be unable to form sophisticated organ systems like the liver, the brain, the spinal column, all these things which are essential in that marine larvae, because of their nature, are not given large amounts of lipid reserves from their mothers, and they're not released at a very early stage of development, a very late stage of development at all. So in terrestrial organisms, there's a lot more parental donation early in life, which allows organisms to not have to consume PUFAs within a few days of birth, so they won't starve to death. But terrestrial organisms have also adapted a number of enzymes which allow them to convert lesser, more saturated fats into polyunsaturated fatty acids inside the body. But this has a metabolic cost and has various maladies when you're doing this at scale. So it's far more efficient for terrestrial organisms to consume omega-3 fatty acids, and it's absolutely essential for marine organisms. So if we're looking at a world where we have to produce more protein to feed more humans, and we need to be farming the seas and utilizing all the surface area, and we need to be producing marine organisms, we need to find a way to produce large amounts of EPA and DHA, because without these in the feeds, we will not be able to produce these organisms, let alone producing sufficient amounts for feeding the human populace directly for its various nutritious benefits. Wild ecosystems have been centered and diversity radiates around sites of mass PUFA production. This map here demonstrates prevailing winds, the trade winds, the most reliable ones that circulate around the world. Now, because of a lot of oceanography and transport, basically the movement of these winds move water from the surface away from the coast in areas of coastal upwelling. So because all this water is rapidly being moved away from the surface, water from the depths, it's much colder, is being moved up. This water has a bunch of nutrients, which then stimulate mass algae blooms. The algae then produce mass amounts of polyunsaturated fatty acids, which radiate upwards to the trophic levels and into all kinds of forage fish and krill, and then all of this is distributed throughout the world's oceans. But it's important to note that primary production and PUFA production occur in anything as small as a rain puddle all the way up into these huge fisheries, which are located in the red zones in the map here. And these red zones are essential to know because these are the sites that produce 90 to 100% of the world's fish meal and fish oil. These are the sites where the ocean's production of golden fats are harnessed within the tissues of oily forage fish like anchovies and sardines and copepods, which then go into these forage fish, and these forage fish are then harvested en masse, and those are distributed. That is how the world has industrially distributed PUFAs, by extracting them from the ocean and then distributing them to the various industries on land. We will be briefly talking about this conventional process of the world's distribution of PUFAs. In this system, natural systems, mostly oceanic in those coastal upwelling zones, produce PUFAs en masse and then accumulate those PUFAs en masse in forage fish species. So this is an ecosystem service that produces the commodity and then renders it into an harvestable form. These forage fish are then harvested en masse. They are then processed in a fish meal / fish oil facility where they have to be processed, gurried, strained, and then all those oils have to be extracted and purified, and then the various byproducts are then divided up into their grades, whether or not it's for a nutraceutical application like the bottom right, so a high-grade application, or something like fish meal be put into your Tetra Flakes. So there's a huge range of products that are extracted from this mass scale extraction, but it is the plain language that nature is doing the baseline producing and primary accumulation and harvesting of the PUFAs. So the issues I have with this system is that it's unsustainable in every sense. It's economically unsustainable because it makes a few key coastal regions fuel the world's intensive lust for fish oil and meal. If you want a hog farm, you need fish oil and meal. If you want a milk fish farm, you're probably going to have some fish oil and meal in there. So you're going to need it no matter what agricultural product you're trying to derive, as long as it's not something that photosynthesizes. And because we have made this such a commodity, and there's growing increased demand, the price for fish meal and fish oil is increasing ever more so. So almost to a point where it will be unsustainable economically to continue to extract these fish, because you'll create a bottleneck where only the people that can afford to buy it will do so. Also, it intuitively leads to the exploitation of the people of those coastal regions, and of that resource itself. It's both leading the population to overexploit their inherent resource, and it simply drives the fact that if prices are increasing, they're most likely going to find a way to charge the fishermen less, and charge the workers, or give the workers at the factory less. So because of this system, there's too much inherent bottlenecks for a baseline commodity, which is important for the nutritional stability of every human being and every agricultural animal and wild animal on the planet. Not to mention that human activity has disrupted seasonal shifts, and even if it hasn't, there are natural events like El Nino events, which can disrupt this coastal upwelling and put a paralysis on these massive sources of PUFAs. And with demand ever increasing, the industries cannot sustain having these paralysis events, and thus having a way to novelly produce PUFAs reliably in the same way that we farm other crops has become more and more of an economic, environmental, and cultural necessity. Every second that those fish sit in the pile, those PUFAs, those lipids are oxidizing. Those precious PUFAs that are only made by nature and not having a substantial alternative as far as mankind producing its own are being wasted. And that's happening with every fish, every harvest. So one can only imagine. And the fact that those fish, at best, are only retaining maybe 10% of the PUFAs that they've consumed in total in their tissues at any given moment. So the absolute waste, considering the pure amount of the commodity which was made by the wild microalgae, compared to the amount that will actually end up in the purified um, fish oil, is staggering. And the fact is, we must find a way to be able to grow the organisms which synthesize these PUFAs and develop novel technologies to extract and purify those PUFAs from those organisms, because this conventional practice is exploitative, wasteful, and criminally inefficient. My research is using the various phyla of microalgae and developing novel cultivation strategies so that they produce high concentrations of PUFAs and other favorable metabolites, and then have high levels of retention of those metabolites, and also to find economic ways to harvest that algae biomass and extract those metabolites of particular interest. So this research is highly complex and will likely take me the entirety of my life. But a few years in, I have noticed a few things. Firstly, algae should be observed as nanofactories, kind of designed by a higher power, nature, if you will. But the fact is that each has been granted through evolution and selection and various other mechanisms such as acquisition, because they tend to eat each other and assimilate each other, is that they each have their own metabolic tool belt, and that they are capable of producing a range of highly unique and valuable bioproducts, but only if the environmental conditions make it favorable for them to do so. So a lot of what I will be doing and what I have been studying is observing how genetic composition interacts with environmental conditions of various algae phyla and how that produces these valuable metabolites, particularly the PUFAs, and then trying to summarize those conditions into practical aquaculture production systems and finding complement novel harvest strategies so that algae can be harvested and produced in an economic way so that it can compete and eventually succeed with the rising cost of fish meal, thus providing a truly sustainable alternative, especially when algae production is further synergized with other industries, allowing for anthropogenic carbon dioxide, nitrates, phosphates, and other waste products to contribute into algae biomass, which can be extracted into highly efficacious nutrition for all of humanity. This is me and the bean during a consulting gig. It's a shellfish hatchery which has various species of microalgae which must be produced in this case, a continuous bioreactor system. Various species here, including Isochryses lutea, Catastris muelleri, Tetraselmis chuai. These species are important because they produce high levels of polyunsaturated fatty acids, which are essential to marine larvae early in life, such as eastern oyster, Crustaceae virginica. The rewards of this research is polyunsaturated production without the need of arable land. You can produce it in the desert or with potable water. You can use salt water, you can even use industrial wastewater if you're clever enough. By utilizing wastewater from other sources, particularly from aquaculture and agricultural sources that have high levels of nitrates and phosphates, and with other industrial sources that produce high levels of carbon dioxide, you can use this industrial synergy to have highly dense microalgae cultures. And by using and manipulating the culture settings, you can have that biomass produce large amounts of PUFAs. And then by having novel harvest and extraction techniques, you can turn that high level of biomass into precious, purified omega-3 oil, which can then be purchased. And more importantly, by having more and more domestic producers offer this product, this oil derived from more sustainable production than wild fisheries, each country will reduce the dependency on those key regions which currently control the vast majority of the world's fish oil production. Now we'll begin to flirt with the nitty-gritty of how to actually begin to plan around this frankly revolutionizing of sourcing of omega-3 oil. The first phase, phase 0, which isn't listed here, is finding the proper algae species and strain, which is paramount for a variety of reasons. And then phase 1, once that species and strain is identified as favorable, is biomass production. So there will be a certain set of conditions which are favorable for increasing biomass of that specific strain, all the way from a few milliliters in a stock culture all the way to a couple thousand gallons in, let's say, a mass tank culture. From that point, there will be a stabilization of the biomass where it's attempted to maintain the biomass at a certain concentration for a variety of reasons. And then a controlled stress will be applied to the biomass. This could be nutrient limitation, excess of light, absence of light, variety of factors, a salinity spike, all kinds of things. Now, it's important that this controlled stress be very accurate and that nothing else is changed besides the specific environmental factor, and that it is non-calamitous, so it does not push the culture to the point where it starts to crash. Now, the idea of the controlled stress is to induce the acquired algae biomass into producing large concentrations of the omega-3 oils, so that in the last phase, that biomass can then be harvested, and then those oils can be extracted. So these stages need not be mutually exclusive, as we'll discuss towards the end. There are technologies that are very promising which allow omega-3 oils to be extracted from biomass. But for now, assume that harvest will require a separate stage altogether. Phase 0 begins with selecting exactly what type of microalgae we want to grow. Microalgae is more a term of human convenience than it is anything of a definitive taxonomic category. In fact, the microalgae not only comprise of different families and orders, but they derive different phylums altogether. So you and a flatworm only have a couple 100 million years of evolution in between you, whereas cyanobacteria may have a few billion years of evolution in between that and that of a more derived microalgae like a diatom. This is increased and made more complex by the fact that throughout evolutionary history, various algae taxa have been derived, it's thought, by acquiring other photosynthetic organisms like cyanobacteria and incorporating them, much like we incorporated our mitochondria. So because of that, each phylum of algae is extremely diverse and has its own set of extreme diversity, and within that diversity, metabolic abilities, some of them extremely favorable. So once you've selected your phylum, you might go down to the family, genus, and then eventually the species. Some that I've worked with and that are particularly favorable for conventional aquaculture hatchery work is the haptophyte T. Eicosapentaenoic latea, produces a lot of PUFAs, Catastris muelleri, diatom, which produces a lot of PUFAs and a lot of other things, and then Tetraselmis chewi, which produces a relatively high amount of PUFAs for a chlorophyte, but also produces a lot of other important nutritional compounds. Then you get down to the specific strains, because each of these species are continuously going through generations and becoming distinctly different. Once you purchase stock culture from a hatchery and you start growing it for a few years, you have a distinct strain from what you purchased. So these strains are then maintained by different facilities, but they are also becoming uniquely distinct within any facility that continuously grows its own microalgae from its own stocks. So examples of specific strains are offered by labs such as the Bigelow Lab up in Maine or the Texas Lab, and they're given different designations. So within each strain of algae, there's an inherent genotype, a different genetic array. Each of these can be expressed or not expressed as dictated by the different environmental factors, but all of this is baseline established. This genetic array is established and can be manipulated and increased through practices of selective breeding, induced mutagenesis, such as irradiating with UV, or various techniques of direct genetic engineering are being developed as well. Gene knockout, gene silence, gene knock-in, chloroplast bombardment, various different techniques for phase 0, establishing the culture that you're going to be amplifying through cultivation and what are its inherent genetic capacities. So something else which kind of mixes in the in-between between environmental and genetic factors are the fact that any genotype has its particular environmental thresholds that can be observed. They can only go to such and such a high salinity or low, or such and such a high temperature, etc. But with each of these different environmental conditions and genetic conditions comes the capacity or non-capacity to accept or reject different symbionts, different bacteria, which can be associated with the outside or the associated water column with the algae. And this symbiotic community can drastically affect the metabolism and culture performance of microalgae. There's thought of having like exactly pure, sterile, just algae cell cultures, and this keeps everything simple from a planning point of view, but at the same time, reality dictates that there will be bacterial colonization, and which bacteria are colonizing and interacting with the bacteria, or with the algae rather, has many dramatic ramifications. At the same time, the different growth phase, the lag phase, the log phase, and then the senescent, and then eventually, or the stagnant phase of algae growth. Different algae cells at different concentrations, and if that concentration dictates the algae culture is slowing down or actively growing, will dramatically dictate both the reproductive rate of the algae and its metabolic activity. An algae culture that is in log phase, for instance, will have a beneficial relationship with certain bacteria, but once it enters senescent phase, those bacteria can then kill the algae culture. Same thing can be done and associated with PUFA production across various different algae species. So if we take a certain algae strain with a certain genetic array and we freeze it in time when it's at a certain concentration in a certain phase of growth, we then see how the various environmental factors exposed to that algae cell at that given period of time then dictate the metabolic activity that we'll have, and whether or not it will be producing or storing or metabolizing PUFAs. So here I have a list of general factors which I've noticed in the literature there is a good predisposition for having beneficial or negative effects on PUFA production. Salinity, in general, more saline species tend to produce and retain more PUFAs. This is, I believe, an artifact of the fact that they do live in a higher salinity environment, and there are osmoregulative ramifications of this. Light. This could easily be an entire talk of its own, so I will choose to keep it extremely brief so it won't go on for hours. In general, blue spectrum of 700 nanometers has been associated with lipid formation. A lot of lipid producers do tend to favor blue spectrum of light, and when they are exposed to blue spectrum, they produce more lipids than when they're exposed to, let's say, green or red spectra. Though it should be noted that a lot of these algae require other spectrums in order to properly contextualize and complete their metabolism. And at the same time, you might be reconciling or compromising lipid production for growth. So there's always room for incorporating and intermingling other spectra. There's also the fact that other species grow heterotrophically in the absence of light and can produce lipids. So that's an entire other factor of thought where light isn't even considered. But for the most part, I have been engaged with photosynthetic microalgae in my studies. Temperature, this is also highly variable. There are several Arctic species that are great examples of extremely high lipid producers, as manifested with the Alaskan natives and their rich, oily fish diet. But there are also warmer species that can also be extremely efficient lipid producers, and they tend to do better in mass cultivation settings, which usually take place economically in, let's say, greenhouses or in big open pools exposed to open sunlight, which tend to get warmer. So these guys tend to grow quicker, stay stable at higher temperatures. pH, this is a whole other can of worms. This dictates carbon availability, but it also dictates a lot of other metabolic functions. The dosing of carbon dioxide is extremely important when you're dealing with heterotrophic algae. There's also the extreme consideration of carbon sourcing and the form of carbon presented. So huge can of worms there. Nitrate, this is a huge one, essential for photosynthesis. And there's a quite huge deal of literature that refers to restricting nitrate in growing algae cultures and having this restriction of nitrate, switch the algae from predominantly metabolizing carbohydrates and then switching over to lipid formation. Nitrate is a good way to stress your algae out without killing it. So they will produce higher amounts of these lipids when deprived of nitrate and nothing else. Phosphate is limiting in freshwater systems, so it's pretty efficacious to limit phosphate with freshwater algae if you want them to produce more lipids. Silicon, essential for diatoms to synthesize their test. It's like a greenhouse that we live inside to amplify light around them and produce drag, and its architecture has various other functions. But to limit silicon can also be a way to deliver stress to the diatoms and have them produce more lipids. And then there are various other vitamins, macrovitamins, magnesium, iron, various other things that can be used to manipulate, be deprived, or release in excess to algae to deliver a controlled stress or to fuel their biomass. Many environmental factors are confounding, and reconciliation should be centered around economic feasibility. All right, so a lot of this stuff interacts, and it's very difficult to say you have this species, this strain, here's this magic formula for lipid formation. At current standing of world research, I believe that very good educated guesses can be made, but as far as fully maximizing the true potential of PUFAs, there's still so much work to be done as far as exploring the genomic potential of microalgae and then producing the most efficacious strains of PUFA producers, and then finding the environmental conditions which most produce those PUFAs. So with absolute efficiency still subject of, the industry when it matures in the distant future, decisions really should be made around economic feasibility. How cheaply can you make this microalgae, and how cheaply can you make it survive and make a decent amount of lipids, and how cheaply can you get those lipids out? So without the industry maturing, we need to have an industry to have it mature. So decisions right now should be centered around economic feasibility. The big puzzle right now is not producing the algae biomass, and efforts to increase the efficiency of lipid production in the biomass is complementary to producing that. Where the real bottleneck exists, in my opinion, is harvesting the biomass and extracting the metabolite of choice in an economic way. So there are various conventional methods of harvesting algae biomass, each with their pros and cons. Filtration is only suitable for the larger species. Otherwise, it is virtually quite difficult and is economically expensive. Flocculation is cheap, but at the same time can produce an unfavorable product for further processing or for feeding off to other organisms, depending on what you're doing. Centrifuging. This is a technique that I've done. It is energy intensive and, again, may produce algae cells which are unfavorable for further processing. Freeze drying can destroy very energy intensive and can destroy lipids. There's also the idea of trophic ascension. So like algae was utilized in antiquity, use other critters which are natural bioaccumulators to do the next step and use their natural adaptations, use their inherent nanotechnology to acquire the algae and convert it into their own tissue, convert those PUFAs into their own tissue, find the organisms that can do that most efficiently so, and then harvest them for oil and meal extraction. So there's also the convention of having the algae biomass converted into a more usable biomass. And then there's milking technology, which I am currently in the process of trying to explore. And this is an idea which surpasses the harvest step altogether in which the algae is treated more as a biofactory and less as a harvested crop per se. But in general, the area of harvest and extraction is the area which needs the greatest innovation and will be the most likely the most favorable and lucrative application of new and novel nanomaterials and techniques. Slightly more details into this process of milking algae for oils. My research into this is still in its infancy, I think it is in general. But the convention is that we are growing the algae biomass and that the algae is storing mass amounts of neutral lipids inside a vacuole within its cell. Now, with conventional techniques, that algae cell would then be harvested, lysed, and then various techniques would be used to extract those lipids chemically. In the same way, I think that it is highly inefficient to take an egg if you're trying to get the yolk and smashing it and then trying to use chemistry and various other techniques to get the yolk back. With milking technology, various biostimulants are applied to the algae so that they are forced to extract and emit the oils throughout their membrane, keeping the algae cell intact and allowing one to maintain the algae culture and allow it to do its own separating on the nanoscale excretion of these oils. In the same way that it would be a lot more efficient to allow the egg to emit the yolk itself and have you just collect the yolk in a drip. In conclusion, the facts on the ground is that the world needs new sustainable solutions for the fact that there's more and more humans on the planet and that we have a dwindling, finite amount of base commodities. We only have so much freshwater, arable land, and we only have so much natural production of polyunsaturated fatty acids. So if we're going to increase human populations, we'll need to increase agriculture and aquaculture. And if we're going to increase aquaculture, we need to find ways to produce PUFAs, most favorably by utilizing the other wastes from other industries, including CO2 and excess nutrients. So microalgae are a classical tool of nanotechnology. They have been used since humans have been around, and they warrant a revitalized exploration in the 21st century. And they have received this. And the rewards of this exploration will be a greater understanding of the microbial world, how algae cells grow, how they interact, and how the nanoscale nature of their existence operates. Because they do operate, they are the largest part of the nanoscale. And then we will have a greater ability through this understanding to manipulate these microbes into biosynthesizing and operating nanoscale properties and complex functions to produce important, valuable metabolites, such as PUFAs, that benefit all of humanity. These PUFAs and metabolites can then be produced at a greater purity, eventually, and without the need of freshwater or arable land, and hopefully will also allow the reduction of human waste as well. So I believe that microalgae, because of their operations at the nanoscale, will become one of the many tools wielded by humanity to usher in a new sustainable age in the 21st century. Thank you for your watch.